There's someone here trying to get this smartboard to go. We're going to ignore but appreciate it. Right, so, okay. So we've been here, we've looked at the general rules for the synthesis of macromolecules. There's also a figure in the book, I think it's figure 210, if memory serves me well. But it essentially tells us the same story. So important to remember is macromolecules are all synthesized from activated subunits. The book uses the term subunits attached to a carrier. And that makes that the addition of subunits is a downhill energetically favored reaction. Okay. So where does the energy come from? There is one important, well, there are several molecules, of course, lots of molecules in cells that contain biochemical energy. But the most important one is the molecule ATP. And this here is the structure of ATP. ATP stands for adenosine triphosphate. And I should translate that for those of you who are true Americans. They can only understand adenosine triphosphate. Right? So, if we look at the structure of this molecule, you can see a couple of important things. And this is a very good opportunity to point out one thing we want to do in this class, and actually in many aspects of biology, is to understand the relation between structure and function. So we see a structure here, and what does the structure tell us about how this molecule could function? Well, let's just look what we have. We have here the 3 phosphates, and we see that if one of this terminal phosphate here, if it is released, then we have a release of energy, which is indicated here as the delta G is negative 7.3 kilocalories per mole. The delta G stands for change, delta, in free energy content, and G is the symbol for free energy. G for Gibbs free energy. Gibbs was a biochemist. And free energy simply is energy that is available to do further work. Now this is the delta G0 prime here. And what that means is that's the free energy change if we would be under standard conditions, which is 25 degrees, pH of 7, and every of the reactants at 1 molar concentration, which is of course not a real situation. So this indicates a certain amount of free energy. To put this into some perspective, the amount of free energy that is stored in glucose is something like 360, 340 kilocalories per mole. Okay, that's 1 mole of the sugar glucose contains some 360 or so kilocalories per mole. 7.3 is a decent amount. Honestly, because of the concentrations that are prevalent in the cell, where ATP is in higher concentrations than the ADP, with only 2 phosphates, diphosphate. Because of that, the real energy content for ATP in the cell is probably more something like 11 to 12 kilocalories per mole. Okay, so there is energy released when a phosphate is removed. That means that when a phosphate is added back to ADP, the diphosphate, to form ATP, the triphosphate, that means that we have to input that same amount of energy. So plus 7.3 kilocalories per mole under standard conditions. Okay, so we have the phosphates there, that's the energy carrying part. What else do we have? We have here a ribose sugar, and there's a lot of stereochemistry. And there are a couple of polar groups, hydroxyl groups, oxygen, hydrogen, on this sugar. And that means that enzymes can specifically recognize this sugar group. In addition, there is this nitrogenous base, adenine, which is a flat two-ring structure. There are double bonds, making this a flat and rigid structure. Once again, there are polar groups that stick out. And so overall, this backbone here, composed of the adenine and the ribose sugar, is a handle which enzymes can recognize. So you can think this functions as sort of a key that fits into locks that are enzymes which have an active site that can recognize ATP. So it's quite a clever design by nature, you can say. Right? We have this handle making this molecule recognizable by specific enzymes, and then we have the energy carrying phosphates. So we see already a relation between structure and function. Now there are other nucleotide-derived coenzymes. ATP is a coenzyme, as we will later see. And many of those coenzymes share this ribonucleotide design in that they have a nitrogenous base, a flat structure, and they have the ribose sugar. So they're very recognizable by enzymes. And this is exactly why most of the coenzymes are structurally similar to ATP and are ribonucleotide derived. Does that make sense? So think always when you look at structures at the relation between structure and function. It helps. So this is a structure you should be able to recognize, and you should be able to tell me what happens when ATP is converted to ADP here, adenosine diphosphate. So I told you when the phosphate is released, there's energy released. Why is that? There are 3 reasons that you should be able to remember. So the first thing is that we have charges in each of these phosphates in ATP. And the charges are all negative. And you know that in electrochemistry, when the charges are the same, there's repulsion, opposite charges attract. So if we think about ADP here, we add a phosphate to it to form ATP, we have to bring a negative charge closer to another negative charge. That is not favored. That would take energy input. So to make ATP from ADP requires energy input because we have to bring the same charges close together. It's not favored, right? So the reverse reaction, the release of a charged part, releases energy, free energy, just like you push in a spring that takes energy. One reason, the charges, okay? But it's not the only reason. If we think about here phosphate, he got it to work. If we think about phosphate, I didn't think it. I didn't. If you think about phosphate, you see there's a double bond here. That's it. There's a double bond, and that double bond can be on any oxygen surrounding this phosphorus. It's a choice. Okay? But there's less choice for where the double bond can go here when this phosphate is attached to the other phosphates in ATP, because this position here is not open. Okay? So there's more opportunity for molecular randomness, we can say, in a DP and inorganic phosphate compared to ATP. In other words, there's more entropy in the free phosphate. So that also means that there is free energy stored in the system. Okay, and then the final reason I've already mentioned, in the cell, the ratio of ATP over ADP is kept fairly high. A good estimate is anywhere from 10 to 1,000-fold excess of ATP over ADP. And that means that the laws of chemical equilibrium favor the hydrolysis of ATP to form ADP. Are we good on that? All right. It's almost a guaranteed exam question, by the way. Okay. Right. So, summarizing for all macromolecules, we have monomeric building blocks, and that means that there's a limited number of defined structures that are possible. The possible structures by proteins are limited by the fact that we have no more than 20 amino acids to choose from. Plenty, by the way. The subunits are activated before addition by coupling them to a carrier, typically something that's derived of a ribonucleotide. Okay. They're added one at a time, step by step. In the overall process, water is formed, therefore we speak of condensation reactions. You should remember that. And polymers can be modified after the subunits have been added together. This does happen in the case of proteins or nucleic acids, even lipids. Okay. Now, in the cases of nucleic acids and proteins, there is also a defined order in which the subunits are added, and there's a defined start point and end point. And there's only one direction of synthesis possible. So for nucleic acids, you always start with the 5' end, end with the 3' end. I can define it in a moment. And for proteins, we start with the amino acid, that's the amino terminus, that has the 3 amino group, and it always ends with the C terminus. So that's the order of addition. And the direction of synthesis and the sequence of synthesis in the case of nucleic acids, DNA and RNA, and in the case of proteins, is determined by a pre-existing structure, which is always a nucleic acid sequence. Okay? So the sequence that we form for RNA and DNA, and even for proteins, is ultimately determined by a sequence given by a pre-existing nucleic acid. And there are base pairing rules that tell us cellular machinery what will happen. Good. How are we doing? Things will get much more exciting soon, I promise you that. Okay. The final thing I want to summarize in my short introduction is the summary of the relation between proteins and DNA. And we can call that the central information processing system. So what do we have? This is the central dogma as it was thought up by Watson, from Watson and Crick, some 50 years ago. That's still pretty much true. So we have DNA contains the genetic information, that is the information to produce proteins, mainly. DNA is copied into other DNA, and that's responsible for the continuity of life, in a process called replication. You should know that replication applies to DNA. Copying DNA into new DNA, that is replication. Right. And that DNA encodes for proteins by means of a messenger RNA or mRNA intermediate. So that process by which we form mRNA from DNA is called transcription, you should remember. And then the sequence of nucleic acids is used to form a protein. We have nucleic acids, we have ribonucleotides in RNA. We have amino acids in proteins, so we go from the language of nucleotides to the language of amino acids. Therefore, it's a translation process by which we form proteins. So translation applies to go from mRNA to protein. I would really appreciate it if you do not confuse replication, transcription, and translation. It's not hard. And it helps. Sometimes it helps. Okay, so not only do we need to have a way to instruct what the protein looks like, we also need some information about where and when in an organism, in which cells and at which time in an organism a protein should be formed. So at the DNA level, the information that is required to form a protein looks something like this. Let's say this is a DNA molecule here. Let's say this is the protein coding sequence part here in yellowish. We need also some signals to tell the cell where and when this gene should be activated for transcription. And that sequence that controls this process is the promoter. We can call that an on-off switch. Okay, if the promoter is active, then the coding sequence with a little bit on the two ends, on the 5' end and the 3' end, is copied into RNA. In eukaryotes, we often see that there also is splicing happening. We'll get to that later. That's a detail we don't care about too much now. And that coding sequence is used ultimately in the process of translation with the aid of other RNA, tRNA, and ribosomes, which is a complex of several proteins and RNA again, to form our protein. That's our protein. That protein folds depending on the instruction in the protein primary sequence. It folds and becomes active. So these things, the proteins, give the cells their functions. So you kind of see a lot of information here, and part of this class is dedicated to understanding how we form proteins, how those proteins work, and how the cell manages to form the proteins it needs at a certain time. And then again, how to get rid of the proteins that it does not need. Alright. So, I've looked back at some of these echo lectures, and it's really scary to see yourself. I know I say too much, all right and okay. I'm working on it. Yeah. The rest of the section 1 slides is used for self-study, but I want to take you through a couple of slides yourself. So the rest of what's in here, you just have to learn, and I think it's not hard. I want to give you a couple of tips. So I want you to understand the structure of nucleic acids and their subunits. And for instance, you need to recognize the 5 nitrogenous bases. If we give them to you on an exam, you need to be able to name them. Now here's a tip. I think everybody can learn, and probably most people know that in DNA we have A, G, C, T. In RNA we have a U instead of a T. People remember that somehow. Most people also can remember that G base pairs with C and A with T. And that in RNA it's A base pairing with U, because there's no T, there's U in RNA. Don't look at that. Now here's a tip about how you can remember the structure of these guys in a more easy fashion. One thing I think that helps is, so we know G and C base pair, but you have to think about the structure-function relation. We're asking this for a reason, and recognize useful structures. If we look at here, I think I should use the cleverness of this board. If I should. Okay. I'm doing my best. Okay, I think now it's captured this. Now, do something else. Bear with me. Okay. It works. Okay. You like blue color. Okay, so C base pairs with G, but how does it base pair? You need 2 electronegative atoms. In biology, that's nitrogen and oxygen, to share a hydrogen. That's called a hydrogen bond. So there's hydrogen bonding between the G and the C, but how? Well, if you look at some other pictures in chapter 2 and some other chapters in the book, you might find that if you take this C and kind of flip it, And you see that this group here really hydrogen bonds with that oxygen, and this hydrogen is shared between the nitrogen here and there, so that oxygen and this nitrogen share this hydrogen, okay, and this hydrogen is shared. Does it work on the big board? It works. Okay, and then finally this hydrogen is shared between this oxygen and that nitrogen. So what we see is there's a business part of this molecule which functions in the base pairing process. A lot of students can remember that the GC base pair is strong. You have 3 hydrogen bonds. So you need 3 electronegative groups that can share a hydrogen. And I can see them here. So if we look at this business part here, you see there's 3 different electronegative groups. That's why it can form the 3 hydrogen bonds. And we see the same in the G. There's 3 different electronegative groups. And this is not true if we go to adenine, which here has a single hydrogen. And there's no electronegative atom there, so we cannot share a hydrogen in a hydrogen bond. So therefore, the base pairing here, okay, is like so. Between the nitrogen here and the nitrogen there, with this hydrogen being shared. And between this nitrogen and the oxygen there. Okay? It somehow works. Now another thing that you might see is that these 3 here, the pyrimidines, are simple rings, and the purines are double rings, are more complicated. So there's a little rhyme, and it goes something like this. Learning about the basis of DNA and RNA is pure agony, purine, agony, AG, agony, pure agony. The pure agony ones are more complicated. I'd rather cut a pi, pyrimidine pi, and cut CUT. And remember that. There's another thing. Okay, if we look at the structure of uracil, it occurs in RNA, and thymine occurs in DNA. Why? Well, it turns out that there's a reaction called the deamination of cytosine. This group here can be removed. That nitrogen goes away, and it's replaced by an oxygen, essentially leading to the formation of uracil. So this occurs. Now, DNA is supposed to be stable genetic material, so if there's an easy transformation by which cytosine can be converted to uracil, then that, since uracil is not found in DNA, if this happens in DNA, cellular repair mechanisms can come into action, because uracil is recognized as something that should be in RNA, not in DNA. So, in DNA, that's the extra methyl group, which allows the cell to distinguish uracil from thymine. See how that works? So the deamination of cytosine can be reversed by cellular repair mechanisms in DNA. Okay, enough. All right, so we go back here. So the remainder of this section 1 we're going to learn. And that means we can now switch to following section. Before I do that, I got all your responses. Someone asked me something that I want to respond to. So someone said that I've heard that this class and biochemistry are similar and cover much of the same topics. Is that true? Well, for the topic I just covered, yes. There's some overlap. There's a lot of material in this class. I think it's more than 70%, which I think is unique to this class. is not covered in biochemistry as far as I know what they do in biochemistry. So that's that. Another thing to say is that if you still need a take-home exam, you can pick it up at the end of the class. If you need an answer sheet still, and you need to turn in the answer sheet and only the answer sheet on Friday, then please do so. And please also understand that you need to treat your answer sheet very nicely, because if you do something like this, then it's not very good in the scanner. So we don't do that. All right, let's talk about how we study cells. How we study cells. Okay. So what do we study? I've already said I'm really interested in the relation between structure and function. So we can have a structure and we can ask the question, what is its function? Or we can have a function, such as, say, respiration, and ask the question, what are the structures responsible for this process? And I think the two are always related and we always meet in the middle. Okay. So, if you come from first looking at a process, you can ask the question, what is the set of components that mediates this process? Think of respiration, the process in which you regenerate ATP in your cells. It serves for giving you energy to do things. They can ask which cellular structures mediate it. And the answer to that question would be a couple of cytosolic enzymes and the mitochondria. What is the minimum set of components? So what can we strip away so that the process still takes place? And then what is the structure of the components that are involved? The mitochondrion has two membranes. What is the significance of having two membranes? What is the significance of the inner membrane of the mitochondrion being so highly folded? The significance, as we'll see, is to have a large surface area. Okay, and then finally, how does each individual component work? And that approach is somewhat similar to the approach taken by the author whose name I always forget. I apologize, I'm sure one of you knows. Or in the book Zen and the Art of Motorcycle Maintenance. Now, this is a book about a guy, sort of a teacher, who I think takes himself and his son on a road trip through America, discovering himself in America, and is going on a motorcycle and is all the time repairing his motorcycle, as it often breaks down. Not too often, which is a good repairman. And the strategy is, he says, okay, what do you do when the motorcycle is broken down or when something's not functioning? Well, you try to find the component that mediates the process in question, and then you try to understand the function of that component, and if it doesn't function, you replace it or you fix that part. So it's kind of like that approach for, yeah, your microphone has died, so is it the battery or is it something else? You take it apart, go to the minimum set of components that you need. For instance, we see here the motorcycle. It has two wheels, it has an engine, looks like it, an exhaust pipe, the steering device, the gas tank, and the body seat. Is there a function for the body seat in driving the motorcycle? Well, how can we find out? If we take the motorcycle, we remove the body seat, can we still drive it? Yes, we can. Will it be comfortable? No, it won't be. By the way, I don't drive motorcycles. I don't know from first hand, but I can kind of guess. On the other hand, there's a chain here connecting the engine to the back wheel. If we remove that chain, the engine will go, the back wheel will not. So the function of the chain is to drive the motorcycle using the power of the engine, on and on. This is an approach similar to what we often do in cell biology. Okay, but there's a limitation, I'll get to that in a second. So we can also look at the structure and ask, what does it do? So we see a mitochondrion. Let's say we have an EM picture, we see all the structures in there, we can ask the question, what is the function of all of that? And then what happens if we change that? There's an enzyme that we think is the ATP synthase in the mitochondrion. What happens if we destroy somehow that enzyme? If we take that out of the system, leaving the rest intact, what will happen? Okay, so that is kind of an approach we take in cell biology. I think much of the time this approach is valid and it is useful. I need to, however, point out a couple of limitations. I need to point out two important limitations. One is that unlike a motorcycle, life is self-organizing. So there's a completely different view of life, which is here. Ooh, there it is. Like what happens with a stream, if we have a rock in the stream. So if we have a rock in the stream, or if I have a stream and I stick a stick in there, you get eddies that form. And these are kind of self-organizing structures. The eddies that form here behind our rock. In the flowing water, those are self-organizing structures that depend on something that we put in there, and they depend on energy in the flow. I'm not meaning to be metaphysical here. But it's important to understand that life is self-organizing. If we damage ourselves, our bodies repair. If we damage our motorcycle, it will not repair itself. It's very important. Another important aspect of life is it's replicating. Life makes more of itself. That is not what motorcycles are doing. So the comparison with machines is limited. That's all I should say. Now, another important aspect of cell biology is that ultimately we want to understand the processes as they happen in living cells. But in order to look at living cells, we have to take them often out of the body. We have to do things to the system by which we disrupt it. So we have to be very aware of the potential for artifacts when we look at our systems. Okay? Let's just keep that in mind. So with all of those limitations, what a cell biologist really needs to do is you need to be a jack of many trades. So we need to be able to study intact cells, maybe in cell culture, or very indirectly in a kind of a way using genetics to study the potential functions of cellular components.